today afternoon speaker, Professor, officially Rosario Rizzuto, but I mean Sarino, I'm not going to talk about Rosario now. So I know Sarino for quite a number of years, I would say by now, mostly through working in the cell death field, where the field of mitochondria was closely touching my area of research. And Sarino is now a rector of University of Padova, which is the top-ranked Italian university and one of the oldest in Europe. He is a professor of general pathology, and he used to be a director of the Department of Biomedical Sciences and member of the University Senate of the University of Padova. He actually finished undergraduate studies in medicine and surgery, if I understood correctly from Italian, which is not really my language. Anyhow, and that was in '86. And he became then lecturer in Padua in '92, professor in Ferrara in 1998, with a short break in States for a few years. And in Padova, he was also director of the Emilia-Romagna Genetics and Biotechnology Network and dean of the Pharmacy School. In 2008, he returned to University of Padova as professor of medical faculty and became university rector in 2015. He was actually elected on October 1st. So his research interest has been centered on the study of cellular signaling, especially on intracellular calcium homeostasis. So just to mention the most recent real breakthrough of his research was a Nature paper in which he described identification of the mitochondrial calcium uniporter, which is the last fundamental component of the cellular calcium signaling machinery which was discovered. And this result opened molecular era of mitochondrial calcium homeostasis that combines molecular insight into the structure-function relationship of mitochondrial calcium uniporter and its regulators, and the possibility of manipulating mitochondrial calcium handling in cultured cells and animal models. So the major goal of that is to clarify its role in physiological regulation of tissues and in the pathogenesis of the most prevalent human diseases, which include a number of neurodegenerative disorders, ischemic heart disease, and cancer. Sarino published over 280 papers in international peer-reviewed journals, and he has been cited, according to Scopus, over 29,000 times with Hirsch index of 90 in Web of Science. Well, I think you should update this data. I just looked today, and I think in Web of Science, which is usually a little bit behind, you have an H index of 92 and around 31,000 citations. But I mean, there is another R. Rizzuto, which publishes in Dance and similar journals. Rachel Rizzuto. Any relationship? No. Okay, so there is another one. Anyhow, because of really fantastic research results and career, Sarino got a number of prizes, including Chiara Donofrio Prize, Biotech Award, Theodor Bucher Medal, and the Antonio Feltrinelli Prize. And he was also elected for an EMBO member in 2013 and for a member of Academia Europaea. So please, the stage is yours.
So I think I have this, so I'm fine. Thank you, Boris, for this very nice presentation, and thank you, Jadran, for the invitation. I will present you I hope a fairly broad seminar in the field of mitochondrial calcium signaling, as the initial picture shows what has turned out to be today the view on mitochondria, which are the organelle that not only provides the most important energy currency, which is ATP, but also a key controller of cell life and death. But I just added 4 slides. I had the chance to be in the previous presentation, in which issues about science and where the science is going, what are the principles of science. So with a bit of pride, since we are also close to an anniversary, we are going to be 800-year-old university in 3 years, I will show you 4 slides about my university. And since, as you heard, I also wear a hat. It's difficult to wear the two hats because being in the lab and running a big university at the same time is not easy. But I want to tell you a little bit about the university, because it really says of what in 800 years, but also today, is the principle of modern sciences, which is, I translate patavina libertas, freedom in research, as an impulse in curiosity-driven research, in addressing the most fundamental questions in science, and not necessarily what will become application tomorrow. And as you know, this is an open issue in funding of science. And I think that if we look back and we think where we are today and what are the principles today, and as you know, we are considered an important university in science, and we are considered an important university in science because the freedom in research, the patavina libertas, has been a hallmark throughout the 800 history. And this is a piece of Brecht's Galileo, in which it claims, maybe you don't know that in Renaissance, when we consider the golden area for culture, for science, in fact was immediately after the Council of Trento. So it was in period in which Europe was sharply divided for religious reasons, and if you were in a part of Europe, you could study only if you belonged to the religion. And there was one exception, and this exception was the University of Padua, that had a very liberal ruler that was the Republic of Venice. It's close to all of us. And Venetians, yesterday as today, they're very practical people. They want to obtain the most of what they can get. And they understood that the university needs to be free if they wanted to get the largest advantage. And so they allowed everybody to study. They play tricks like Italians sometimes do. So formally, they could give the degree only to someone who declared himself Catholic, but they decided that the body that would control that would not be the bishops, but would be a university body or a state body, and of course they would never control. And as here are one of the most important scholars, which is Galileo Galilei, when he was fleeing to Florence, the rector, my predecessor at the time, says, don't do it, because remember that some prince offer more money. But here we can offer freedom. We are a free university. We are a university where even Protestants can become, and I think this is a value that explains why the opening and the full opening to everybody and to all views, this is something we are also very proud about, is that the first woman in Earth graduated in Padua. And this is very nice to say. What someone should remember is that this happened in 1678. So it has not been for, again, the Renaissance, the golden era of Renaissance. Women could not graduate. Now we have 60% of our graduates who are women. But the first woman ever was in 1678. It is something. And of course, we're very proud that it happened in Padua. And of course, freedom in science, you know that the great strength was not only that people could come, but they could do science. And this is the oldest anatomical theater in the world. You have several copies around Europe that were the exact perfect copy of the one of Fabrizio Acquapendente. And something that I say to our guests when they come and visit this old botanical garden, which is the oldest botanical garden in the world, is that it's beautiful. It's a UNESCO site. But people should remember that it was a laboratory. It was a hortus simplicum. It was a garden of medicines. This is why it was, so in a sense, it was an old pharmaceutical laboratory. Now we see it as a beautiful place, but this is what it was born. So let's go back to mitochondria. What are mitochondria? And you all know that mitochondria, this is how they were originally seen and described. Mitochondria means threads and knots, because if you cut a cell, you find some mitochondria that are elongated and some which are cut. And this is where they get the money. This is one of my old pictures, and this is a three-dimensional high-resolution rendering of mitochondria. And you see that they are a very dense network within the cell, largely interconnected in a regulated manner. And people who work in mitochondria know that the unity of mitochondria, fusion and fission, is a tightly controlled phenomenon that has a physiological consequence. And as to function and how mitochondrial function is regulated, this, as you know, has been the great contribution of Peter Mitchell, who demonstrated that mitochondria, this old endosymbiont arriving into the primordial cell, capable of utilizing oxygen for producing ATP. It does it because it generates a transmembrane proton gradient, so that intermediate, there was no phosphate intermediate as it was looked for decades. But the real intermediate is the pumping of protons across the ion-tight inner membrane, and this huge gradient, and we'll come back to the huge gradient and why this is relevant for calcium, is the driving force that runs the energy-consuming reaction of ATP production. And you can do this experiment in vitro. You can isolate mitochondria, and as soon as you give a substrate, you see that they make a membrane potential. How is mitochondrial function regulated? So this is common textbook knowledge for students. This is what every student should know, what is their structure and how they are regulated. But how are they integrated into cell function? What signals do they receive, and how are the signals decoded? And of course, as Boris was saying, I have been throughout essentially all my career investigating the most common second messenger of living cells, which is calcium. We all know that through a variety of channels in the plasma membrane or in the endoplasmic reticulum, calcium is transiently increased within the cell, and the increase of calcium in the cell is translated into operation of the cell. And you have some examples. This is a contracting cardiac myocyte, and you see a calcium waves every time the muscle contracts. These are neurons, and you see a calcium rise going through the neuron as soon as an electrochemical or a neurotransmitter is fed to the cell. Do mitochondria participate in this calcium signal? And here I come to the final result, which is, of course, yes. Otherwise, I will not be presenting the seminar today. And you see that, that if you image again the calcium rise, you see that you have an increase in calcium in mitochondria when a cell is stimulated. So this is the final answer. But in fact, the story of mitochondria and calcium is a very complicated story. And this is because the idea that mitochondria, energized mitochondria, can accumulate calcium is a very, very old concept. You see a paper of 1961 in which it was demonstrated that if you energize mitochondria, so if you give substrates to mitochondria, and then you give pulses of calcium to isolated mitochondria, every time you give a pulse of calcium, calcium goes into mitochondria. You see, calcium goes up and then turns back down. And it does it a number of times till the mitochondria cannot do it anymore. And this phenomenon attracted a lot of interest in the last 20 years or so, which is permeability transition and the fact that mitochondria, when they are overloaded with calcium, at some moment they are not able to accumulate the calcium anymore. But look at this part of the trace. Mitochondria can rapidly accumulate calcium. And of course, this generated the concept, and this was the concept of the 60s and 70s, that mitochondria play an important role in the signaling I described before, in calcium signaling. Why was that? It was because it was demonstrated that mitochondria can have some transporters. Very cautiously, they were called uniporter and antiporter. So only calcium goes in or calcium goes out in exchange with another ion across the impermeable inner membrane. By that time, Peter Mitchell had demonstrated the chemiosmotic theory, so it was clear that the mitochondria have a very impermeable inner membrane. And what was demonstrated at the time was that mitochondria can rapidly accumulate calcium whenever the calcium oxide mitochondria rises. Why is that? Because, as I told you before, the respiratory chain generates a membrane potential which is largely an electrical potential. So 180 millivolt, a cation like calcium has a huge tendency to go in. Actually, equilibrium would be reached only for 6 orders of magnitude higher calcium in the mitochondria than outside. So calcium has to go in. And so it was clear and discussed that calcium inevitably would go into mitochondria. But then came the 80s, when the field of cellular signaling exploded, and 2 fundamental concepts emerged. The first is that the calcium rises that I showed you before, so calcium increasing in the cell, does not originate from the mitochondria, but originates from the endoplasmic reticulum. Again, every student, first-year biology students, knows that when a cell is stimulated, calcium is released from the endoplasmic reticulum. But this was discovered in the 80s by the work by Mike Berridge and Robin Irving. And the second concept, which further killed the notion that calcium would actively move in and out from mitochondria, was that the transporter of mitochondria, the so-called mitochondrial calcium uniporter, is a low affinity transporter. Is a transporter that would work only for concentrations which are in the orders of tens of micromolar, hundreds of micromolar. And the living cell will never have 100 micromolar in the cytoplasm. So the concept in the 80s, when I started working in the field of calcium quite extensively, was that mitochondria were an irrelevant organelle in intracellular calcium homeostasis. The concept was they have the potential, maybe it's serendipity, maybe it's an organelle that will become relevant only when the cell is overloaded with calcium, in toxic calcium overload. Of course, not a very exciting perspective. If you read a review on calcium and signaling, mitochondria would be the final paragraph that essentially stated we don't know what mitochondria do in the calcium signaling field. And this is where I started my scientific career. And I started my important scientific career. And I started my important scientific career because I joined a laboratory that was very active in calcium signaling, in the field of calcium signaling. And my successful idea was that if you know that calcium is measured with chemical dyes, but if you want to be very precise in measuring calcium, I proposed that a chemical dye was not the best way to measure. The best way to measure is targeting a protein to a specific, a calcium measuring protein to the organelle of interest. You may say that this now is common in the GFP era. This is absolutely common sense. There is nobody that ignores that you can target a reporter protein to a specific site in the cell. But this was the first targeting of a measuring protein, a luminescent protein or a fluorescent protein to an organelle. And fortunately, it worked. And when I thought of this idea, I said, we will start from mitochondria. And we will start from mitochondria for the simple reason that mitochondria have a very simple intracellular targeting. You put a mitochondrial pre-sequence, a sequence that has certain characteristics, and every protein will go to mitochondria. So I thought this will be a proof of principle. We will be able to show that you can target a reporter protein to an organelle, and then we will do it for the important organelles. Then we will do it for the endoplasmic reticulum, that we will do it for the subplasma region. This will just show nothing. And of course, science also often gives you the results you don't expect, and the results we didn't expect was that when you stimulate a cell, well, first of all, the targeted protein goes to the right place. So this chimeric protein of a pre-sequence and a luminescent protein measuring calcium goes where the pre-sequence dictates, which is to mitochondria. And when we use this reporter protein to measure calcium within the mitochondria, and this is a typical calcium trace, this is a non-excitable cell stimulated with a mediator of inflammation, histamine. And you see that what we know is that there is an increase in the green trace in the cytoplasm. What was totally unexpected was that the mitochondria would also have a rise. Actually, you see that the rise is much greater than the cytoplasm itself. And this was showing exactly the opposite that we and everyone could expect. And of course, the first explanation is, no, all the data, previous data were wrong, but there is always a better way to investigate an unexpected result. And to make a long story short, what we could demonstrate is that mitochondria have a low affinity system. So in principle, they shouldn't sense 2 micromolar, because for 2 micromolar, they shouldn't accumulate calcium. But the mitochondria, they're not floating in the cell, but they are strategically located in close proximity to the channel. And this is what I show you in this, one of the evidence we give in this slide. And you see that if you label, by the time it was 1998, GFP had arrived, we could label organelles with GFP. And you see that here, labeling the mitochondria and the endoplasmic reticulum with 2 different colors. Red is the endoplasmic reticulum, green is the mitochondria. Calcium goes out from the endoplasmic reticulum and goes into mitochondria. If we make a high resolution of the 2 organelles, you see that they are very, very close. Oops. And when you go in detail, and here you eliminate the second image, which is the image of the endoplasmic reticulum, there are some areas of the mitochondria in which the mitochondria is exactly in the same space, is exactly in the same place, because they are physically attached to the endoplasmic reticulum, which means that the mitochondria respond so efficiently because they sense microdomains, they sense a local domain, which is very, very close to the endoplasmic reticulum. They do so because ATP production is regulated by calcium. I will not enter into the biochemistry of this, but there are 3 key enzymes that are regulated by calcium, and therefore, when you measure ATP concentration, by that time we had fun in targeting the protein, so we targeted an ATP measuring protein. We could see that this correlates with ATP production. And so we were starting to put in place what the mitochondria do and what calcium does into mitochondria. And this is a very simple way to say that when you stimulate a cell, you are triggering some energy-requiring processes, and the mitochondria produce more ATP. So the fine-tuning of metabolism to the needs of the cell is operated by calcium. Calcium goes in, calcium stimulates the enzymes, and this produces more ATP. But then comes cell death, and Boris and the field that the calcium is, so the idea that calcium goes into mitochondria for the simple fact of stimulating metabolism turned out to be a simplistic view, because calcium in mitochondria can do something completely different. One was fairly simple. If you put a lot of calcium in mitochondria, an organelle that has a negative potential, you start to cycle calcium in, out, till you kill the mitochondria. And this is clearly what is shown here. This is an overload of calcium, such as the one that happens in an ischemic heart when you reperfuse. And you see that if you do so, you have, you see here you're measuring membrane potential, then at some moment you see membrane potential collapses, you have a complete overload, and the cardiac cell dies. This is what we would call dramatic death or necrosis. But then we learned that cells die not only by necrosis, not only because they have an accident and there's something dramatic happens to them, but death is a continuous process of living cells. A body requires continuously a cell, some cells to die because they're not necessary or because they're dangerous. And you need to have a very quiet way to die. that in a poetic term was called apoptosis, such as the drop of an old cell from the plant. Who discovered the phenomenon didn't call it apoptosis. He called it shrinkage necrosis, one way of necrosis. But now we call it apoptosis. And is the physiological or physiopathological way of eliminating unnecessary cells. And of course, this is very relevant for pathology because in some cases you have too much cell death. In some cases, cells escape from death, such as tumors do. And mitochondria, you all know, play a fundamental role in this because a regulator of apoptosis is released from mitochondria, and when this happens, you see this is cytochrome C is released from the mitochondria, and then the cell undergoes the dramatic process of cell death. And calcium plays an important role because when you combine an apoptotic agent with a calcium increase in the organelle, you have the induction of cell death. And this is, I will not enter in detail, a pro-apoptotic protein, which is a protein of hepatitis B, causes a calcium overload in mitochondria, and this causes the alteration of mitochondria and the swelling and cell death. And so the final scheme, mitochondria are fully back in the, not only they are fully back in the field of calcium homeostasis, but they are considered a critical site where a decision is made. Because when you stimulate a cell, normally this would serve the purpose of stimulating metabolism, and of course this is relevant because when this is less efficient, you have loss of tissue function with age or in some genetic conditions. But there are a number of challenges, physiopathological challenges that may hit a cell together with the stimulus, and under those conditions the effect is not that of stimulating metabolism, but rather to trigger cell death. And of course, as I showed you just now, this is also obviously quite relevant for pathology. For this reason, the process that I have been studying happened to be gradually more and more of interest to the scientific community. And if you do a search in the literature of mitochondria and calcium, you see that for a long time you had a number of aficionados, never missing in the literature. But then after we demonstrated that that calcium goes into mitochondria physiologically, this plays a role, there has been a drastic increase. And if you look at the numbers, we are talking about 2,000 papers per year. If you type mitochondria and apoptosis, you get 3,500. So we are in the number of one of the topics that are very, very strongly interesting for the scientific community. And of course, we missed the channel. We missed the channel. It was relevant, it was important, but nobody knew what the channel was. And I can assure you that at least in the past, after the publication of the paper, many groups have searched. I've been reviewers of a number of papers claiming that they had found the mitochondrial calcium reporter. In fact, this has always been very unsuccessful. And the reason why it was unsuccessful is that in science, we tend to be cautious. And cautious means repetitive. We tend to do things as the others do. And if you want to identify an unknown protein, what do? Either you have an inhibitor, you make it radioactive and you isolate the protein, but there was no good inhibitor. Either you go through genetics, and we have a fantastic genetic system, which is Saccharomyces cerevisiae, but unfortunately, fortunately for me, but unfortunately for those who have searched, Saccharomyces cerevisiae doesn't have the mitochondrial calcium reporter, or you do it in silico, because you search proteins that resemble the protein you are interested in. In this case, very simple. You look for all the channels of the plasma membrane or the endoplasmic reticulum, and you look for proteins that are similar to it, and you need to find it. And as you will see, it's a totally different channel. Don't forget, mitochondria were bacteria. They came in, and they are completely different from the repertoire of the rest of a eukaryotic cell. And so, fortunately enough, nobody found it, and we decided that we would go through a bioinformatic approach, but an bioinformatic approach led by scientific biological knowledge. So we said, we will look into a database of mitochondrial proteins, so a gene set of mitochondrial proteins, which is available. It's called mitocarta. There are approximately 1,000 genes of proteins that are for sure mitochondria. And we will restrict the search gradually based on what we know about the mitochondrial calcium importer. And we know that it is present in every cell, so it has to be ubiquitous. We know that it is a channel, so it needs to have a transmembrane domain, a hydrophobic region. It needs to be absent in Saccharomyces. The reason why they fade is that it's not in Saccharomyces. And it needs to be present in lower eukaryotes that we knew had a mitochondrial calcium accumulation. And nicely enough, if you follow these 4 constraints, you make 1,000 possible genes to get down to 14. Then I like always to say that we're Italians, so we are a bit lazy, and 14 was a lot. I mean, it was too much. We couldn't cope with 14. I mean, it was very, very difficult. So we said that even among these 14, we wanted to prioritize. And this was very important because, of course, we are facing competition, so we needed to be fast. And we prioritized by looking at the transmembrane domain of these 14 proteins. And of course, we looked for a protein that had a very strong consensus, a very strong retention of the transmembrane domain. If it is a channel, you cannot mutate it. It has to be. So there was one candidate that stood out because that candidate has a very strong sequence conservation in the transmembrane domain. And this protein with a name that said nothing, coiled domain C109A, turned out to be the mitochondrial calcium importer. This is just to say that we needed to do a few things to get the paper published. I mean, one of the comments after the first revision was, the paper is solid, convincing, but there have been so many failures in this field that we want the authors to do it. And I was tempted to answer, it's not my fault. I mean, why should I pay for all the mistakes that were made? But we were asked to do the experiment in reconstitution, lipid bilayers, mutagenesis, blah, blah, blah. It turned out that it is the right protein. And so this is a courtesy of a colleague and friend of mine that quite nicely puts this timeline in which 50 years after, exactly 50 years after the demonstration that mitochondria accumulate calcium, we have the molecule. What do we know about this? What can we do with this molecule now? Well, first we can test all the hypotheses. I told you that calcium overload leads to cell death. Now we can do it. We can overexpress a channel that puts a lot of calcium in the cell. We can put it in a cultured neuron, and of course, when you introduce the mitochondrial calcium importer, they die more. But what is nice, you can do it in vivo. So you can inject a virus that produces more mitochondrial calcium importer, and you see that when you do so, the brain of these animals, the neurons die more, as exactly what we were predicting. But of course, it's nice to see. What do we know now about this mitochondrial calcium uniported? Well, we know that we have been terribly lucky. And we know that we've been terribly lucky because the system is molecularly very, very complex. There are there are 2 proteins, 2 alternative proteins that form the pore channel, the one we identified, and an isoform. And there is a complex regulatory system, which is called the MICU1. So why have we been lucky? Because we were lucky that simply by expressing the pore function, if the pore required this additional protein that we discovered later for functioning, we would have had the right protein in hand, but never find it out. So we've been fairly lucky. We know now, well, we have all the molecular details. We have also made the reconstitution and the structural analysis. We know that it is a tetramer. In the tetramer, we see the pore. You see, very nice. We see the 4 proteins creating an ionic pore in the center. We can see the top where the regulators are, so very, very nice. We know a lot about this protein. We know that there are 2 alternative forms of pore protein. I will not enter in detail, but just pass you the concept, which is important, that the two proteins that can form the channel, they're not equally efficient. One of the two is very good, one of the two is fairly bad. And depending on how you mix this, you have an efficient channel or a less efficient channel, as these slides show. Why is this relevant? Because the tissues express different amounts of the two. So if you have a tissue like the heart, where calcium goes in every second, because our heart beats every second. So every second, you have an increase of calcium in the cytoplasm of a heart cell. You don't want a super efficient system. Otherwise, you will have an overload of calcium in mitochondria, and you will kill the mitochondria of the heart. And nicely enough, you see that the non-efficient subunit, the B, is highly expressed, while the very efficient subunit is not expressed. Conversely, skeletal muscle is a tissue in which at some moment you operate. And when you operate, you want to be very efficient, and then you produce a lot of the hyper-efficient subunit and less of the less efficient subunit. But let me show you one example, because also before cancer was cited, and it's not so easy to the idea that you can tackle cancer so easily, because cancer cells continue to mutate and continue to mutate by being as efficient as possible for the state in which they are. And we know that a metastatic tumor, when the tumor originates, the most efficient struggle it has to face is not to be killed. So it must resist apoptosis, it must resist cell death. When they become incredibly aggressive, the real need of the cancer is to run away. The real need is to look for energy, to look for food. And therefore, it's much less important to be resistant to killing than to be able to move. Otherwise, the simple fact that you're growing will kill you. So, and nicely enough, if you look at, remember the two forms of the pore, the very efficient and the less efficient. And you see that when a cancer becomes highly metastatic, the most aggressive forms, these are breast cancers in which that you can stage in different levels of aggressiveness. And when they become very aggressive, they produce the channel that makes a lot of calcium to go in, counterintuitive. They will become more sensitive to cell death. But they do. And why do they do so? Because when they have a high calcium rise in the mitochondria, they are very rapid in moving. They become very, very capable of metastasizing both locally and systemically. And so they face the risk of being more sensitive to killing cells, but they can unfortunately expand very much. And of course, we could also demonstrate it, because when we silenced the MCU of a very aggressive cells, you see that the capacity they have in vivo to spread is much less. And I will conclude by showing you in a few minutes one example that puts all the complexity together. And the example that puts all the complexity together, why you have very complicated molecular system to do one simple thing, which I think is the problem we are facing as biologists. And skeletal muscle is a very good example. Skeletal muscle has very large increases of calcium in mitochondria. The electrophysiologists know, the biophysicists, they can measure, and they measure that skeletal muscle has huge currents of calcium. Why do they do so? Well, first explanation, because they have a lot of the efficient subunit and very little of the inefficient subunit. And maybe we will be happy with this. My colleagues were not. They wanted to find out that there was an additional reason. The additional reason was that the regulator has a splice variant. The splice variant is expressed only in skeletal muscle, and therefore the regulator becomes a super activator in skeletal muscle. But I will not enter in detail, except by saying that you have two different molecular mechanisms by which skeletal muscle selects to have a super channel. Why do they have a super channel? And so we did an experiment in vivo, and we introduced either the gene for expressing or the system for turning down the gene, two different ways, electroporating or virus. Of course, when you introduce the gene, you have more calcium going to mitochondria. What happens if you put a lot of channel for calcium? What was unexpected was that if you do so, the muscle becomes hypertrophic, becomes a large, strong muscle. Of course, if you do the opposite, if you reduce the amount of calcium, there is a reduction. And how does it happen? And here we discovered something we were not expecting, because the first explanation that you would give and that I would have given is they are metabolically more efficient. Calcium stimulates metabolism, and this is why they make the big muscles. Not at all. If you overexpress, metabolism is exactly the same. If you reduce, if you silence, you have less metabolic activation. But that is not the explanation. So what is the explanation? And the explanation, again, I will not enter into the biochemical details, but take the line. What happens is that when you overexpress the calcium channel, the signals to the muscle for becoming hypertrophic becomes very efficient. This is not a trivial concept, because we are discussing about an effect of mitochondria on a nuclear process that controls the growth of a muscle. So this means that there is a signal that from the mitochondria, from the old bacteria that is there, and we said it's there to produce ATP, then we said no, it's there also to kill the cell. Now we are saying that the mitochondria are there, and they continuously communicate with the rest of the cell to the point that they are saying to the nucleus, we want a very nucleus-specific process. We want a large muscle. We want the muscle of an athlete. So this is, in the field of mitochondria, something we should, I think, work more and more on the mitochondria to nucleus signaling. Mitochondria are talking to the central control of the cell. And they're talking not only because they provide ATP, not only because they activate the cell death processes, but they continuously exchange signaling molecules that makes the cell makes strategically unexpected decisions. And I will conclude by showing you the data on the mice that are exactly the same. Fortunately, genetics allows us now to make not a cell, but an entire animal, and an entire animal in which we eliminate a gene. But not only we eliminate a gene, we eliminate a gene only in the tissue we are interested in, so that we have an effect. If we are looking at skeletal muscle and calcium in mitochondrial skeletal muscle, we can eliminate MCU only in skeletal muscle. So if something happens, nothing happens in the liver, nothing happens in the brain. It's specific. And when we do so, we see exactly the same. Of course, in this case, we have a full animal. So we can look whether the animal is efficient or not. As you may expect, he is weaker. He cannot run very extensively. But if you look at why this happens, of course, it doesn't take calcium in the mitochondria anymore, as we may expect. And when we look at the pathways, we see exactly the same thing. The dimension of the fibers is reduced. The pathways for hypertrophy are inhibited. Of course, he runs less. And I will not, but of course, in the area in which, as overfed populations we are interested in, which is metabolism, we also realize that this has a big impact in the use of substrates. And therefore, what we see is that on the metabolic side, these animals completely change the type of substrates they utilize. They utilize a lot of lipids, and they utilize much less glucose. And my final slide is the slide in which I hope I gave you at least some hints of how we are in, I think, in a very fascinating biological era, in which out of a process which is a process of great relevance in signaling and great complexity, which is how calcium goes into mitochondria, in the last 8 years, because all this happens, as Boris was saying, in 8 years, because the molecule, this molecule was identified, the first molecule, in 2011. Now we have learned that there is a huge complexity. There is a huge molecular complexity, and our body in the different tissues and in physiological or pathological conditions continuously changes the composition of this toolkit. It's like Maurizio is an engineer. They are engineers as well. They continue to change. They take away a piece, put another, and modify it a little bit. And you have a continuous, highly flexible complexity. And of course, what is the function of this complexity? That fortunately for calcium in mitochondria, we know how this correlates with regulation of metabolism, regulation of cell death, regulation of waves of calcium in the cell, and therefore control of the signal of the cell. And of course, the challenge that we have now is to understand how molecular complexity and cellular mechanism becomes the way in which we have the full physiology of a normal person, so how development happens, how organ physiology is regulated, and of course, what happens in disease, what happens in genetic diseases, what happens in environmental diseases, and what happens in the vast majority of our diseases, which are the diseases in which our genetic gene set and the environment mix together and decide what will happen to us. And with this, I thank you. Of course, this is a slide that every experimental scientist has to show, because what we show is what younger people do. But as you may understand, for me, it's particularly important to show it because I don't know how much time Maurizio has, but I reserve myself one day in the laboratory, one day. And it's also very difficult to preserve that day in the laboratory. So if the work goes on, it's because these group of people are very talented, very enthusiastic, very, very active. And of course, this includes senior people like Diego, Cristina, Anna, and Georgia, and the younger people that essentially they coordinate and they participate in the time that they have. And I thank you.